Okay, now we'll talking about the second lectures on the viruses are the step of lysogenic life cycle, temperate phage as an example. So in the first lecture, we talked about the life cycle of the viruses, where we talk about the lytic induction. In the lytic induction, we discuss about how the viruses are harming the host cells and released out from the host cells after the killing. So this is called the lytic induction. Now, in case of lysogen, the cell, that is host cells, adopt the virus particles or its genome inside it to get integrated, and this we call the temperate phage or the lysogenic stage of the phage particles. Now, in this case, attachments and entry of the phage particles takes place through, again, injection of the linear double-stranded genome. When they get entered into it and that integrated into the host genome or host chromosome. Circularization of a genome takes place, the 12 base cohesive ends of the lambda DNA takes place. We call the at P site, or in case of bacteria where it get integrated, we call at B site. So this is called the, through recombination process, we call the site specific recombination takes place in integration of the phage DNA into the bacterial genome. The early proteins, because certain proteins which are there which suppress the basically the phage genome to be activated independently, or rather it get integrated into the host machinery through certain proteins which suppress the induction process or the early transcription of this mRNA. So these proteins are C2, C3 and CRO protein and N proteins which are synthesized. Now these proteins are basically the suppressor also. It acts as a suppressor for the transcription process. For example, if C2 is attached, or C2 is stable, we call protected by C3, we call the lysogeny is favored. It means the lytic induction is stopped. It suppresses the lytic induction. And if C2 is degraded, then CRO dominate, it means the lytic induction begin. So the lytic induction is basically, so if C2 is degraded, then CRO dominate is formed, and this CRO dominates, which is called the suppression of the lytic cycle enzymes, and so the lytic cycle start, induction begins, takes place. The role of C2 and integrase. The role of C2 and integrase is basically C2 activate transcription of INT gene. INT is an integrase gene which helps in the integration of this virus into the host genome. An integrase which catalyzes the recombination between the phage and the bacterial genome. And this we call the at sites, we call at B on bacteria and at P on the phage site, between GAL and bio-operon. Now, what is integration? Integration is the phage genome integrated into the host genome, all right, and then it becomes prophage, then it maintains the lysogeny. Now, how the lysogeny is maintained basically? What is the process of maintenance of the lysogeny? As I have told you in the earlier slide, the C2, C3 and CRO proteins, they are involved in maintaining the lysogeny or in induction of the lytic cycle. So how does it work? Let's see. So C1 is we call the lambda repressor protein. So C1 is expressed. When C1 is expressed, it suppresses the lytic genes, that is CRO, Q or lysine genes. Now these lysine genes are lysogenic genes are lytic genes are suppressed by the by the C1 suppressor, but it activates it into the own promoter we call PRM. Now the prophage DNA is replicated along with the host genome, and the host genome during the cell division. So when it allow the host cells to grow, to replicate, unlike the lytic phage where the host genome is suppressed. So when a host cannot multiply, the cell remain static state, and the number of virion particles are formed in the cells, and it kills the cells, lyse the cells, which is not in the case of lysogeny. The stable lysogens are called the long time stable lysogens are called the dormant stage of the bacterial cell, or the dormant state of the host cells, where bacterial cells survives for long and long time, years and years and years it can be, and it divides normally. Cells can divide normally along with the bacterial and the phage DNA together. And each daughter cells may inherit the prophage. So it means every bacteria produced after the lysogen state of the phage particle carries the phage DNA. And at any time, any bacterial particles could have induced under the certain stress conditions and form the number of phage particles within that bacterial cells. Induction is called the exit from the lysogeny. So for example, so this is the bacteria and this is the bacterial genome. I'm sorry, I don't have another pan color. So this is, I'm making different structures of the genome. So this is the phage genome, and this is the bacterial genome. Oh, in the phage genome, this is in the lysogen state, or temperate phage. Now this lysogen state at temperate phage, when induced, we call induction. Then this bacteria may carry bacterial genome, and the viral genome get out from the bacterial genome, and then it form the large number of bacterial particles, phage particles inside it, like this. And then this is the lytic stage, and then lytic takes place, the cells get lyse, and number of phage particles are released out. So this is the induction cycle of the bacteriophage. Now, taking this in view, that is how the lytic induction takes place, what are the conditions which cause the lytic induction, right? This is important to understand. So, it triggered by the DNA damage, maybe UV light, some mutagens, maybe spontaneous mutagens or maybe induced mutagens. Both mutagens are conformed. Then host REC-A become active. So REC-A is a protein which cleave the suppressor protein, and this suppressor protein will then allow the various genes to activate, right? And then it causes induction process. It stimulates the cleavage of the lambda suppressor, right? We call the lambda suppressor of C1, because C1 suppressor says this is the gene, C1 suppressor is here bind. This is not allow the various transcription process to move mRNA and to form caps. So this process is basically stopped. Now when the process is stopped through the suppressor, what REC-A protein do, it cleave this like this, and then transcription process which was blocked here is stopped. Is again started, and capsid formation takes place and the virion particle starts. So this process, induction through REC protein or through different mutagens, is basically responsible for the lysogeny to lytic induction. Now lysogeny to lytic induction is basically taking place under the stress condition or with the REC protein, as I have told you. Now without C1 transcription of lytic induction like XIX, cis, int, cro, q resumes. It means when the C1 is removed, the suppressor is removed, all these genes which excise out the integrated genome of the phage particle, right, taking place, and then the genes like a cis, SIS, which called excision gene, then we have int gene, then cro gene and q gene. All these genes get expressed after the C1 protein is cleaved out. Now, the excision process of this viral genome is basically responsible for the formation of the virion particles and lytic inductions, and lysis takes place in the cell, and huge number of virus particles are released out to produce more virion particles for next infection into the bacterial genome or in bacterial cell. So this is we call a switch from lysogeny to lytic. We call the lytic induction process. So once the excised viruses are removed, re-enter into the lytic inductions, replication starts and host lysis takes place. Now this is just to show you some pictorial presentation here, how the process takes place. This is the INT gene and XIS gene, which regulatory genes, and this is the site where the bacterial genomes process integration takes place. These are the C3 genes, N genes, CRO, and C2 OOP genes are present. Now these genes encode the product involved in the regulation process of the virus, lysogenic to lytic induction, are the lysogeny cycles to incorporate. So there's a two type of cycle. Don't get confused with this figure. We have a two. One, either the phage particles go to the lytic induction if C1 proteins get expressed, or if it goes to the lysogenic state, then all these genes which responsible for the excision is get suppressed, right? Or they allow them to go for the temperate phase or lysogenic phase. Now try to understand the N-terminator, which we call the N-terminator gene. These N-terminator genes allow the C3 and X-axis genes to again form the transcription mRNA of these genes, and all these C1, C2 genes get allow the INT gene and cis genes to form. Now when the INT and cis genes they form the lytic induction, because the virion particles genome is get excised out from the bacterial genome. Now this is beginning the lytic pathway. So this is a pathway which undergo the lysogeny, and this pathway begin the lytic induction, right? So just it's a play around the role of C1, C2, C3 and CRO proteins and several other genes which are involved in maintaining the dormancy state of temperate phage, or the phage which can completely integrate into the bacterial genome and does not allow it to enter the lytic induction. But sometime when the induction takes place, either through RECA protein to remove the suppressor, C1 suppressor from lambda suppressor, or through some mutagens or UV radiations, which is also kind of a physical mutagen, they can remove it. So this is the basic process of how the decision making process takes place to establish the lysogeny or the lytic phage. So this is how this process is basically involved in the decision whether it go to the lytic induction or it go to the lysogenic temperate phase. Now the generalized life cycle of the HUPS. So here we are discussing of some of the viruses which are animal viruses, are commonly formed like HUPS simplex viruses, HUPS viridae family, and these are the viruses where so there's a difference between the cycle of the human or animal viruses to that of the bacterial viruses. In case of the human viruses, the phage particles, unlike phage particles, the whole virion particle get entered. So this is how the this is the virus particle attached, absorbed, attachments, absorption takes place. After attachment and absorption it takes place, it undergo the uncoating process. Now uncoating process means when the virus get entered, it get uncoated, that is the removal of the nucleic acid, whether it is a double-stranded DNA or single-stranded DNA or whatever nucleic acid types these viruses may have, they are uncoating. So uncoating is the first step of the life cycle of the viruses, right? And this is the process where you can block the virus proliferation or virus infection. Now if you block the uncoating process and make the virus remain in the coated form, that is capsid formations. Uncoating means removal of the coat, removal of the capsid, capsomeres, right, or removal of the envelope if it is enveloped viruses. So dissolution of the envelope, removal of the capsid and removal of the capsomeres are basically allow the virus to uncoat, and this uncoating process is basically the removal of the nucleic acid. Just like in case of lysogenic phase, we deintegrate or we excise the phage particles. So what is important here is the nucleic acid to be removed out from the virus particles. so that the nucleic acid can undergo the replication process. So this is how it get here. The nucleic acid get enter into the nucleus in cell, host cells, and this is the cytoplasm, and there the process of the replication start. This is the concatameric DNA, and this is how the budding takes place, and you have a number of virus particles they are start synthesizing, and these virus particles they are forming the Golgi apparatus, protein synthesis takes place, and this is again vesicles are formed, and through these vesicles again these viruses are exocytosed. Now in case of animal viruses, not always necessary that you have complete lysis of the cells. You can have a number of virus particles that are formed and they exocytose through the membrane itself. So this is the process where without harming the host cells they removed out, but they damage the cells to some extent. Now the attachment of Cy174, which is very common virus, it binds to the specific receptor of the E. coli. This is the phage, right? And then genome entry takes place. The virus double-stranded DNA plus strand they inject into the host cells, and capsid remain outside, right? And this is the example of phage DNA, right? The earlier one example of the animal virus, this is the bacterial viruses, where the conversion of the replicative form, that is host polymerase converted into double-stranded DNA, and double-stranded DNA we call RF, reverse transcription factors, right? And the transcription starts, RF reverse factors serve as a template, mRNA serve as a template for the synthesis of mRNA, and this double-stranded DNA which is formed through reverse transcriptase enzyme, we call reverse transcription, right? An enzyme present within the virus genome, right? And they form the mRNA formation, and this mRNA is again ready to form the DNA and to form the nucleic acid for the virion particle to again. So the process of basically this is called the genome packaging, and genome packaging, it's a newly synthesized double or single stranded DNA genomes are inserted into the procapsid, and the procapsid they form further matured to form virion particle after the maturation process. And then host lysis release of particles takes place with the same mechanism as we discussed. The viral genome E blocked the peptidoglycan synthesis, and via inhibition of MRAY enzyme, which helps in weakening the cell wall, and host cell lysis takes place, and then release of progeny, that is a number of virion particle as a progeny which are formed or get released out from the bacteria by cleaving the bacteria. Now, see, this is how the cycle takes place. This is the DNA plus DNA. This is the bacterial DNA polymerase, and then again you have a rolling ring replication takes place with the DNA, replicative form of the DNA comes, and then transcription takes place. Again the replication process goes on, and then mRNA synthesis takes place, the transcription, they are translated into the protein formation. This is the new virion. Now this protein and this, so there's a simultaneous process of the genome formation, right, as well as the protein formations, so that the virion particle, when they mature, they get the genome, DNA as well as the protein. So this is how the replication starts from this side to this side, and this is how the protein synthesis takes place. Now at the end, this protein and this DNA, they get together to form like this virion particles. So this cycle is integrated cycle of the new virion particles takes place. This is the pictorial presentation where the plus minus DNA, they form mRNA, and the plus minus DNA again. Now this plus minus DNA is important because this plus minus DNA is required to get integrated into the virus genome. This RNA will not get integrated into the virus genome, right? This will change the characteristic features of the genome. So the bottom line is that the viral genome during replication get integrated or get incorporated, assembled into the virion or capsid or capsomers to form virion particle is remain intact as a genome of its own, whether it is a plus to minus or minus to plus. So this is called the multiplication strategy of the double-stranded DNA virus. Now the example of RNA viruses, the multiplication strategy for the double-stranded RNA viruses, where you have double-stranded RNA viruses, you form mRNA, from mRNA to form proteins, and then proteins, again you have this remain back to mRNA. Now this is important to synthesize. This is what I'm trying to emphasizing you. If this is not synthesized, so same genome is required. This is the same genome is required to get incorporated into the virus particle, right? If this genome after synthesis of the mRNA to protein will lost, then the virion particle will not incorporate it with the intact genome. This is the intact genome. So whether it is a double-stranded DNA, double-stranded RNA, plus single-stranded DNA, single-stranded RNA, whatever genome the virus particle may have, should be incorporated of the same form. So on one side, you are synthesizing the genome, on the other side, you are synthesizing the proteins, that is capsid, capsomeres are envelope proteins if they are glycoproteins, lipoproteins, and then they combine together to form virion particles. So this process is called assembly or maturation step. Now again, you have here the plus RNA genome. In case of plus RNA genome, you have again the protein forms and again minus RNA form because it transcribed, and then from transcription of mRNA minus mRNA again to form the transcription of the newly synthesized mRNA. Now this plus mRNA genome is ready to incorporate. So again, whether it is a RNA, it's DNA, or whether whatever, it formed the intermediate transcript to form protein synthesis, that is a capsid, capsomeres, nucleoproteins, right? During this process, simultaneously at the later stage of the transcription and translation, in the cycle formation of the virus cycle formation, that genome is again reconstituted. Now, if that genome is not being reconstituted, then the genome will not, the correct genome could not be incorporated into the viral particle. And that viral particle is may change its nature. For example, plus strand or minus strand to form plus strand, or plus strand to form minus strand to get incorporated. But that minus strand is not required for the synthesis of the protein. For the synthesis of the protein, what you need is a plus strand. Right. So plus strand form protein, but the plus strand again form minus strand in order to incorporate it into the bacterial, into the virion particle before it get infect to another virus particle or to another replication of the virus particle to the new host system, right? Now the life cycle of the poliovirus, this is again example of one of the animal viruses, polioviruses, where the attachments, this is the attachments takes place. See the membrane get engulfed or phagocytosed inside, and they release here the RNA. When the RNA is released, it forms mRNA, that is minus RNA form, and the genome is again formed, plus RNA genome is formed for incorporation into the virus particle. Similarly, this RNA is also formed, translating this transcript into the various proteins. Now, on one side, you are synthesizing the RNA from minus strand to plus strand. On another side, you are taking from this plus strand to form again the proteins, and these proteins, viral proteins, along with the viral RNA, again composed or constitute a new viral particle with the correct genome and with the capsid protein. So this is how the beauty of these viruses cycle, whether it is animals or whether it is plant viruses, whether it is bacterial viruses. So this is how this virus get removed out from here, right, to give the virus particle out from the membrane through lysis. Now the negative strand RNA virus again. Say in case of negative strand RNA virus, again the same thing I can explain you. The minus strand cannot form the protein. It cannot form protein. So in order to form the protein capsid, you have to convert into the minus strand to plus strand first. So this is called a plus RNA, right? And then this plus RNA to form the protein, and then from protein to this. Now this process again this minus RNA, they form plus RNA, and then from plus RNA they form the minus RNA. Now, why this minus RNA is formed? This minus RNA is formed because the original form of this virus is minus RNA, this one. So, this, until and unless you have the original genome formed through this life cycle of the virus particle, the virion particle will not form. So, in order to form the virion particle, what you need is basically to reconstitute the intact virus particle with the same viral genome. Now, the life cycle of influenza viruses. Again, this is a little busy slides. Font may be small. I'm sorry for that, but I think just to look at this picture. See, this is the coated pit. It means where the coated vesicles which are formed from the cell is generated, and this vesicle at pH 5 to 6, they release the RNA. So this process we call the uncoating process. Right. Uncoating process means suppose this is the capsule and this is the capsid course, and this is, say for example, this is the virus particle. Say I can take the whole like this. So this is the RNA, or this is the virus particle. Now this virus particle carrying RNA. So when we call uncoat, what we need is, say, this pen drive which I am using as RNA or DNA or nucleic acid, it gets removed out like this. It uncoating. This is coated, this is uncoating, and this is released out. Now, this is the DNA, RNA, or any nucleic acid type of virus, depending upon the type of virus, is ready for the replication process or for the transcription process of the life cycle of the viruses. Similarly, uncoating takes place, and this uncoating takes place with the help of the host machinery. Different enzymatic, different pH conditions provided by the host machinery, and enzymes are released out which can function at that pH to lyse the coat and to remove the RNA or DNA in that case. So, and then this RNA get penetrated into the nucleic acid in the nucleus. Now, why the nucleus? Because the nucleus is carrying all the machinery for the host cell to replicate, right, to transcribe and to form protein in the cytoplasm after it get released out from the nucleus after the transcription. So then mRNA comes out from mRNA is formed in the nucleus, and the mRNA come out to form protein synthesis. Now this process of the cycle of the influenza viruses, which is single stranded RNA viruses. Now at the end of the cycle, the single stranded RNA viruses, see, this is an incorporation of the single stranded RNA viruses takes place in the virus particle. So when the virus particle they form, using the host machinery, they form capsid, they form capsomeres, large number of capsid and capsomeres, and then at the end they release out from the from the host cell after incorporating the single stranded RNA viruses. If single stranded RNA virus could not be incorporated, in that case what happened? The correct RNA genome is not part of the virus and the virus cannot survive. So this is the example of retroviridae or retroviruses. Now in case of retroviruses, say RNA plus minus DNA, because reverse transcriptase involved in the transcription process of the DNA, and then double-stranded DNA is formed, and this DNA they form the again RNA through transcription, and this protein incorporated again in the virus genome. This is the plus RNA. Now the only difference between retroviruses and other viruses is that in retroviruses the reverse transcriptase is present, and this then double-stranded DNA get incorporated into the host genome DNA. So whenever there's a induction takes place, and this is the example of retroviruses like HIV viruses, the virus particle can go to the latent stage for years and years. So therefore, the HIV patients which are having HIV infections can survive for many years, and they are not even showing any symptoms. So without symptoms, dormant stage, they can survive for n number of years, 2 years, 3 years, 10 years, depending upon the immunocompromised state and the induction of these viruses. Say, this is the example of HIV, which is one of the example of retroviruses. So this is the nucleocapsid, blue one, the nucleocapsid, then integrase, reverse transcriptase enzyme, which help to reverse transcribe the DNA from RNA to DNA, and genomic DNA, RNA, and then viral DNA, and then these are the Gag ball proteins which are involved in the transcription of these viral DNA. Now, when these Gag 122 proteins present, which are receptor protein, which allow the virus particle to get enter inside the host cells, and then this process goes on. Endocytosis takes place, then receptor binding takes place, uncoating of the virus particle takes place. Now, in case of uncoating of the virus particle, which is taking place here, is basically responsible for the integration of the viral genome. Now, when the removed this and they can lyse out from the cell, before that they get disintegrated or they get excised out from this genome and to form the same life cycle of the virus genome as in earlier cases, and all this budding, assembly, maturation takes place, and finally it gets released out from the cell. Now, the gapped genome of the Hepatinaviridi hepatitis virus HB, hepatitis virus V, belong to the family of Hepatinaviridi, is reverse transcribing DNA viruses. Now, the virion is 42 nanometer spherical particles. Genome is circular double-stranded DNA. One strand is completely but nicked, means like slightly nicked, like here, and then it's 42 nanometer spherical particles. Now, the complementary strand is in complete gapped, and genome encoded the 4 particularly overlapped open reading frames we called ORF, which transcribe into the new genes and translate into the protein. Now, infection occurs mainly in liver cells because it's a liver virus, hepatic cells are involved, and because receptors are present on hepatic cells, the hepatic cells can only incorporate these viruses, no other cells can incorporate. So, infection takes place only in the liver cells, right? And transcription by the RNA polymerase 2 generate the multiple mRNA copies, and this multiple mRNA copies are basically allow the number of RNA virion particles are formed, and so from a single RNA, single virus particle is formed, and from virus particle we can have replicated so many other virus particles to make n number of the virus particles form. So, this is how the whole viral genome works, and the take-home message is that the 2 types of life cycle of virus particles are there. One is called the lytic cycle, another is called the lysogenic cycle. In case of lytic cycle, the virus particle go into the genome, degrade the genome, and use of bacterial genome or the host genome to form number of virion particles within the cells and lyse out to kill the cells and they infect to the other neighboring bacterial cells. In case of lysogenic stage or the temperate stage, whether it is animal viruses or bacterial viruses, where the integration of the integration of the virus genome into the host cells takes place, and this integration basically prolong for as long as it undergo induction process through the expression of several protein like C1, CRO, and other proteins which may act as a suppressor. RECA protein may also involve in the suppression of the RECA C1 proteins for the cleavage of these repressor proteins for the induction process. So, the induction from the lysogeny to lytic is called the induction process, which again forms. So, ultimately the process of the virus particles maturations and release takes place only and only after the induction process takes place. With this, I thank you very much, and I think this is the good book you can consult as a reference book for studying this virus particle. Thank you very much.